Okay, so we left off last time with introducing the idea of how we can make proteins, so how our bodies and cells make proteins from our DNA. Okay, so remember we talked about the structure of DNA and that if we break it down to its most basic structure, it's a really long strand of codes using 4 different letters. And we said that DNA is so important to our body that we can't risk having that DNA move from the nucleus out into the cytoplasm where the proteins need to be made. So where we're headed now is the process of manufacturing these proteins. And this can be divided into 3 main sections. And we're going to spend probably a day on the first 2 and then another couple days on the second one, or the third one, sorry. So the first main category of making proteins, or where we begin with making proteins, is something called transcription. So I'm going to bring you back to this diagram that I showed you yesterday. And here we have just a basic cell. And inside of our cell we have our nucleus. And housed inside of our nucleus is our DNA. And we said we know the DNA has to get out there, or at least the message of the DNA needs to get out into the cytoplasm in order to make our proteins. And it's too risky to let that DNA molecule go out itself, so we need to first make a copy of it. And making a copy of that DNA so that that copy can travel out into the cytoplasm, that process is called transcription. Now, in the process of transcription, a few things are done to that transcript. They call that copy that's made a transcript before it can travel out into the cytoplasm. So a few protective mechanisms are done to this before it travels out, just to kind of protect it from any harm that would be done, because still this is an important messenger that has to get out there. So remember from our note on the different types of RNA, one of the types of RNA we talked about was called the messenger RNA. And this is the transcript, or when we transcribe our DNA, this is the copy that gets sent out into the cytoplasm, which is why it's called the messenger RNA, because it's going to deliver that message of what the DNA code is in order to make those proteins. So right now, this note's going to focus on just the transcription process. How can we take DNA, make a copy of it so that that copy can go into the cytoplasm? Okay, so the first part of transcription is, where does it occur? It happens in the nucleus. It needs to happen in the nucleus because we want all of these safe protective mechanisms that we're going to put in place happen in a safe place before we send it out into the cytoplasm. So the genetic code located on a gene of DNA, remember a gene just represents a certain portion of DNA, is transcribed and copied into mRNA. And you might be asking yourself right now, why is it just one specific part of the DNA? Well, your body needs different proteins at different times depending on its own needs and what it's doing. So each of the sections of DNA represent these different proteins. So it's only going to need what is required. And so that's why specific areas of the gene get transcribed, which is another fancy term for just making a copy of it. And that copy is in the form of RNA, and mRNA to be specific. So there are 3 parts to this process. And those 3 parts can be broken into 3 main headings. Initiation, which means to start something. Elongation, which is the building part of it, making it longer. And then termination is a fancy word for stopping. So let's maybe just add those in. Okay, so I just went back and I typed these in because my screen recording video maker was covering up my buttons. So initiating means to start, elongation is the part where everything's getting built, and then termination means to stop. So during the initiation process, you might be asking yourself, how does it know where to start on our DNA? So there's an enzyme called RNA polymerase. Remember, A's, ending of A's means that we're dealing with an enzyme. This smart little enzyme binds to a site that we call the promoter site, and it recognizes this site by this. This is really important here. It recognizes it by the high number of adenines and thymines. So remember, these are 2 nitrogenous bases that we talked about. In your segments of DNA before a gene begins, so different segments of the gene, you're going to have an area where you have a high concentration of A's and T's. And I'll tell you why. If you recall from the structure of the A's and T's, when an A and T bond together, remember that there's just a double bond between them holding that together. But when we have cytosine and guanine, there's a triple bond that holds these two together. So you might ask yourself, well, why is it going in this area? Because if you think about most things in our body, including enzymes, like to do what's easiest. It's easier to break a bond that's just a double bond than a triple bond. So this smart little enzyme knows to look for this spot that's got a high concentration of A's and T's, because this enzyme's job is to break apart that spot to split the DNA. So much like the DNA replication process you guys looked at. So I had you guys look at the replication fork diagram, where there's specific enzymes that go in and help to pull apart the DNA and bind at the DNA, and then there's another enzyme that will read the DNA and know how to build it. So this is exactly what's happening here. This is a version of replicating our DNA into what we call an mRNA transcript. And so the enzyme that we're talking about here is RNA polymerase. It wants to break apart where the double bonds are because that's easiest. Once it's broken apart, it's going to read along the DNA molecule, and it's going to build its new molecule from the 5' to 3' end. Okay, so remember we talked about how each end or strand of DNA that comes together, there's always a 5' end and a 3' end. That has to do with where the hydroxyl group, what carbon the hydroxyl group is bonded to on your sugars. And it's always opposite on each strand, right? That was the term anti-parallel. So we build our mRNA transcript in the direction of 5' to 3', and then it'll reach an area that passes near the end of the gene that needs to be transcribed, and we call that the terminator sequence. And we're going to look at this in a little bit more detail. There's actually codes that these enzymes are looking for that will tell it can stop now. The terminator sequence means that it's signaling that you can stop. We don't need to make any more copies of anything further along this DNA molecule. So these are the three main things that happen when we're transcribing that DNA or making a copy of it into mRNA. So here's sort of a diagram of what's happening. If we have our double-stranded DNA, so each side, remember they run anti-parallel to each other, so one end that's 3' will be matched up with a 5', and same at the other side. The RNA polymerase is going to come along, and it's going to help to find that region of A's and T's, so this would be it here. Oops. Let's do this. Show that we had double bonds holding these guys together. So it's easier to break these double bonds up here and then begin reading. And so what's happening here is in red, this is showing what's being built as a result of reading the code along the DNA strand. And you can see it builds this mRNA in the direction starting at a 5' end in the direction of 3'. So this is the direction that it's being built. And you'll notice that there's complementary letters that will help make your new mRNA transcript as a result of the letter that you're looking at on the actual DNA. So that's something we're going to get to a little bit later. We're going to understand, well, how do we know what letter's matching up here? We're going to get into that in the next step, because we're actually going to pretend we're at RNA polymerase and read through a DNA strand and make that complementary strand based on what we see, because there's a code that helps you figure this out. So right now, all I want you to focus on the fact that we split apart our DNA in an area where there's a high adenine and thymine concentration, and we construct our mRNA transcript in the direction of 5' to 3'. Okay, so once that our mRNA transcript is built, it needs to travel out into the cytoplasm. And before you want to send anything that's important, and this mRNA is important too, out into the cytoplasm where it's going to be coming in contact with a whole bunch of other things, you want to make sure it's protected. So first thing that's going to happen is something called removal of introns. So on a piece of DNA, we have regions that are non-coding, so they're made up of letters, but they don't code for a specific protein, so they call that non-coding regions. And the term for that is introns. So they've labeled the term introns for areas of DNA that don't code for anything. They're just letters that are there. Now, when we have areas of DNA in those genes that do code for things, so code for specific proteins, because that's what we're doing with our DNA. They call them exons. And so before we send that mRNA out into the cytoplasm, we can remove the areas that we don't really need. If an intron doesn't really code for anything, it's going to go through and let's get those, remove them and get them out of that DNA transcript. So introns must be removed prior to leaving the nucleus. And there's a small little molecule that will come in, and its job is to cut out the introns, and they are called spliceosomes. So they'll actually take the actual part of DNA that doesn't code for anything out, and then it will join the remaining pieces together so that all you're left with is one long piece of exons, so the important area of DNA that actually codes for that protein. So that's the first thing that happens before it leaves the nucleus. The second and third thing are more to just protect it. At the 5' end, we put a cap on it. And it's just an extra set of codes that get put at the 5' end just to protect it from some types of enzymes that will sometimes digest and break down that DNA. So it's just sort of an extra piece of protection on the head or the 5' end. And the same thing happens at the opposite end, the tailing. So capping at the 5' end, tailing at the 3' end. And this is another enzyme, poly A polymerase, will add on the tail anywhere from 150 to 200 adenines. And that's again for protection at the other end. So basically what we have is, just to sum things up a little bit, we've taken our DNA, we've split it apart in the region that we need. So the proteins that we're looking for, it reads along there, it figures out what gene we need to transcribe. It makes a copy of that X section of the gene. And then before we want to send that out into the cytoplasm, we want to remove the areas that don't mean anything or don't code for anything. So introns get removed, exons get joined through this molecule called a spliceosome. And then the final process before we send our mRNA out into the cytoplasm is protecting it. So we put a bunch of nucleotides at the beginning or the 5' end. We put a bunch of nucleotides at the end. The capping at the 5' end isn't anything in particular. It could be any one of the 4 nitrogenous bases that get put at the cap. But for tailing, it's very specific. And the enzyme might help you remember that. The enzyme is called poly A polymerase, and that's because it adds adenines to the end. Okay, and these two are protective mechanisms. This is to just get rid of the garbage or the junk that we don't really need before we send it out into the cytoplasm. Okay, so that kind of sums up the idea behind transcription and the RNA processing. So we've made a copy of our DNA, we've protected it, we can now send it out into the cytoplasm to go further into the protein synthesis process. And that's it. I have a little video I'm going to have you guys watch as well.